Our next speaker, we have Chen Wu, who is an associate professor at UCLA in the Department of Computer Science and a research scientist at ByteDance.
Behind the other desktop.
Oh.
Okay, now it's coming.
Oh,
It's coming.
oh, it's coming now.
Oh.
Yeah.
No, you probably have to go here.
Oh, here. Yeah, behind.
Yes, yes.
All right. Okay, great. Yeah, thank you for inviting me, and it's my great pleasure to be here. So in this talk, I will introduce one of our ongoing project, trying to build a foundation model for Cryo-EM data. And this foundation model is based on flow matching and also diffusion transform. So this is a work. I'm currently taking on leave from UCLA and leading a AI for science team at ByteDance. And this is a joint work with my colleagues at ByteDance. So there are tens of EM data, especially the density and also 2D particles generated in the past 2 decades. And you can see that the cumulative number of entries released, for example, EMDB is increasing rapidly. With the rapid growth of the data, the utilization of the data is still under exploited. And in the meantime, generative AI has made a lot of progress in terms of learning the knowledge from different data, and also then use the model to generate new knowledge. For example, for language model, large language models, they try to, such as ChatGPT or Cloud or Google's Gemini, they try to learn the data of text by doing next prediction. And for image generation, models like ImageGen or like a stable diffusion, they are trying to learn the image diffusion by iteratively denoising those noisy images from noisy images to clean image, or mapping the noise to image. And the underlying models include diffusion models and flow matching. So a natural question is, given the huge and rapid progress in generative AI, is it possible to bring those advances in generative AI into the field of Cryo-EM so that we can build some foundation model for Cryo-EM data? And this is our goal. So in my team, we recently are working on a foundation model for Cryo-EM. We call it FM. So here, FM, you can understand it as a flow matching or foundation models, or even diffusion transform. So it's a very interesting name when we came up with this name. And for those of you who are not very familiar with generative AI, especially diffusion models, so let me give you a very big intro to diffusion models and its variation called flow matching. So roughly speaking, diffusion models try to learn to generate data from noise. So starting from some, for example, Gaussian noise, the diffusion model will iteratively denoise the random noise and gradually map the noise to some image, for example, image of cat or image of, for example, dog. And in this picture, you can see that on the left-hand side, there are the distribution of the data, for example, diffusion of the image. And on the right-hand side, they are just the random white noise. And the diffusion model actually, by doing forward diffusion and to first add noise to the image until the image become like a total noise. So this is called the forward diffusion process. And the backward diffusion process is using some thing derived from the score function derived from the forward diffusion process to actually it's like an inverse problem. So given the noise, it will just follow the first trajectory to generate the image. And by training the diffusion model, we are essentially just estimating the score function of the underlying data diffusion, so that once we have the score function by 3D lemma, we can show that we can generate the image from the noise. And on the other hand, the flow matching actually can be viewed as a variation of diffusion models, and because it directly match the trajectories, those trajectories which connect the data and the noise. So in principle, it can be much faster in terms of training than diffusion model. And when we apply flow matching, the more advanced version of diffusion model to Cryo-EM data, so we need to first design the model. We use the diffusion transformer, which is a backbone model used in a lot of the image video generation models, such as stable diffusion tube. And for data, we just collect and curate the data set from EMDB, which is probably the largest public data set for EM. And we did a lot of like data cleaning and pre-processing, including scaling, normalization, and you can find the details in the appendix of the paper. Oh, by the way, so this is a paper which has already been published in iClear, last year's iClear, and we are currently on the journal version of this paper, which try to expand the model to more like real world application scenarios. And the last thing we need to decide is the object function, which is what's the object function for training. And in this scenario, the object function is just the flow matching training objective. So by putting all these three things together, then we could train the first version of the foundation model, CryoFM1. And you can see that similar to image generation. So similar to image generation, we try to do like a lot of like flows or trajectories, which connect random noise to different like 3D density maps of the Cryo-EM data. And you can see that for different, for example, protein, they have different 3D structures. And this is more challenging than the 2D image generation. The reason is, as I mentioned before, the flow matching model actually is trained using a diffusion transformer. But for transformer-based model, we know the attention mechanism usually has like a quadratic compute complexity with respect to the, for example, the size of the image. But for 3D density data, instead of dealing with like 2D like pixels, we need to deal with a 3D voxels or follow cell. And this actually significantly increase the compute complexity from quadratic to the image size to like a cubic in like a 3D like a volume size. And this is the biggest challenge when we train the CryoFM foundation model. And in order to deal with this, tackle this challenge, we propose a technique called hierarchical diffusion transformer. So here's the idea of the hierarchical DIT. So from the left-hand side, the 3D, the like density map of which this 3D cube, which enclose the density map of the protein. We first do, for example, downsampling or some kind of a pooling to reduce the size of the 3D cube. And then we are doing something called a local attention. So instead of doing attention to all the voxels in the 3D cube, we only do attention with respect to a small neighborhood of each voxel. And we repeatedly do downsampling and local attention several times to get a very small cube, which is sitting in the middle. And for this small cube, we can do global attention using the standard global attention in transformer. And then we do everything in the reverse order, because we first do like a down projection, and then we do up projection to recover the 3D cube. And the output of this pipeline actually is just the vector field, which is served as a central component in flow matching. And in this way, we greatly reduce the GPU memory use, so that it will enable us to train foundation model with a lot of 3D densities. And the next question is, we not only want to do unconditional sampling, we also want to apply our foundation model to different downstream tasks. And in order to do that, we use the idea of the so-called posterior sampling. So what does posterior sampling mean? So for the unconditional foundation models, we are essentially learning the score function on vector field, which is the gradient of the log density, and the gradient is with respect to the data x. And by the base formula, we know that the posterior of x given y actually is equal to the marginal density of x times the conditional probability of y given x. And if we take log and then take gradient with respect to x, we can arrive at this equation. And given this equation, you can see that this log p of this p of x is a prime, and we just calculate the score of the prime distribution. And this term actually is like a forward operator. It's a conditional distribution which is derived from the forward operator. For example, given the density x, we have some observation y, and this observation could be, for example, some like a 3D structure, or even like 2D particles. And this actually is defined, and it also is associated with the downstream tasks. And for each downstream task, there's a physics informed process which models the likelihood term. We call this likelihood term. And on the left-hand side, that's the posterior distribution. And by combining the prime and the likelihood term, we are able to get the posterior. And from the posterior, we can do sampling. All right. So to further understand this process, let me give you a synthetic case. So this is a case called the deblurring. So we have some original density, and the further process, or the underlying like a physics process for blurring is we first do some like Fourier transform to get the frequency information, and then we will zero out some outer area in the Fourier space. For example, after Fourier transform, we get this 3D cubic, and then we just only retain those data within this ball, and we just zero out all those information outside of the ball. Then we get this one, and then we do inverse Fourier transform, and you can see that the original density has a high resolution, but after doing this filtering in the Fourier space, actually we get some blurred density. The resolution of this one actually is lower than that one. And this is the blurring process. And given the blurring process, actually we can define the, using the posterior sampling technique, we can define the posterior distribution, which is given the blurred density to generate the original more clean density. So from this example, you can see that for different downstream tasks, there's an underlying physics process which we know how to define the forward operator, or we can estimate the forward estimator from the data. And then together with the prior distribution learned from the foundation model, we can do posterior sampling. And this posterior sampling will enable us to do deblurring. And the deblurring process actually is illustrated using this green dashed those arrows. So from those blurred density using the posterior sampling. So we do the backward way to get the original, to get the estimated clean density. All right. So to put everything together, so this is the prior sampling. So the animation takes a little bit of time. So from XT, which is the noise, we do iterative like denoising to get X0, which is the data. And to illustrate in a better way, so the trajectory of each flow matching is just from Gaussian noise to some sample data. But at this step, we haven't introduced the likelihood. So it's just the unconditional sampling. So if you change the Gaussian noise, then you get a different 3D structure or 3D density. And for posterior sampling, it's more complicated, but it can still be illustrated in a very clear way. So we have some likelihood, and this likelihood is defined based on the observation data, for example, the blurred density. And during the flow posterior sampling, we are doing two things. So because without those likelihood, we just do unconditional sampling, or we call it prior sampling. And given the original observed data, then we can do posterior sampling. At time t, we first have xt. And using the prior sampling, we can get x hat 0. But we haven't incorporated the observational information. So how to incorporate the observation information? So given x hat 0, we can do a forward like a process. We can do a forward operator, for example, FIP followed by some masking. But this masking actually will only retain the information outside of the ball, because the information outside of ball is estimated from x hat 0. And for information inside the ball, we just use those information derived from the blurred density. So from the observation, we do FFT, we do masking, and we have this green colored ball. And by combining this green colored ball and this purple-ish like cubic, which exclude the ball, we get this information. We get this one. And for this one, we do inverse Fourier transform. We get another. We will obtain another like x hat 0. And this x hat 0 is different from this one. Maybe actually use x hat 0 prime, because this contains the observed data likelihood information. And from this x hat 0, we can continue this process to get xt minus 1. Okay, so this is an iterative process from like total like Gaussian noise and to some data. And at each iteration, we will incorporate the observed information in terms of likelihood. Okay. All right, and this is also some illustration of how this flow posterior sampling works. So the input could be the Gaussian noise and the observation, and the output is the sample data. And the observation could be some blurred density. And after posterior sampling, will somehow deblur observation and to get some density with higher resolution. Okay. All right. So in the remaining time, I will just provide you some experiments. So I will go over like 4 experiments, and all of them are synthetic tasks. So for the first task, we call it spectral denoising. And remember that for each downstream task, we need to define the underlying physics process for the forward operator. For spectral denoising, the forward operator actually is defined in this way. So we first do a Fourier transform of the original density to get the corresponding signal in the frequency domain. And then we just add noise. And this noise actually are isotropic noise to the signal in the frequency domain, which is absolute noise. And to apply this to some specific density in the EMBB data set, for example, EMBB 18549. So this is the original data. And after we apply this spectral noise, we get the degraded density, which is illustrated here. And you can see that, for example, the density in this box actually is very blue. And after we do posterior sampling using CryoFM, we will get a high resolution density. For example, if you look at the same region, there are more details in this blue rectangle. And we also compare it with 2 other baselines. One is the deep enhancer, the other is EM ready. So both of them can also enhance the degraded density a little bit. But the enhancement actually is not as good as our approach. And to give you some more quantitative way or quantitative comparison, so we also do the FSC plot. And this light blue curve is the FSC of our approach. And the other curves of different colors are for baselines. You can see that in terms of the FSC, our approach is also better, not just visually better, but also quantitatively better. The second, this is some more like a quantitative metrics, for example, in terms of fail rate. So for fail rate means if the model generates some density which significantly deviates from the ground truth density, then we consider it as a failure. So the smaller the fail rate, the better the performance. And we also calculate the FSC, the area under the curve, and also the FSC at the 0.5 cutoff. And for the other, the second and the third metric, the higher the better. And for different section in this table, actually it's just correspond to different noise level, which will give you a different resolution of the density map. And for each row, this is the metric after degradation. And then we apply 3 different approaches, including our approach. And you can see that our approach can achieve the lowest failure rate. So basically there's no failure. And also the highest FSC metrics. Okay. And we also tried, for example, the anisotropic noise instead of adding anisotropic noise in the frequency domain. So we will add some non-uniform noise in the frequency domain. And you can see that from the visualization or from the FSC plot, our approach is also better than other baseline approaches. So here's another table. I'm not going to explain it again. So basically, we demonstrate this. We use the same performance metric to compare different methods, and our foundation model-based approach gives you the best result. And the third synthetic task is the missing wedge restoration. So basically, just delete some like frequency or information in certain angle. And again, our approach can give you better results than baselines. And the last synthetic task is ab initial modeling. I think in previous talk, in the talk before the previous talk, they also studied a similar task. And for this task, we compared, for example, our approach with CryoSpark. So we just use CryoFM to do ab initio from, for example, several input class averages, just some averaged higher SNR 2D images. And you can see that by using our approach, we can achieve comparable performance with the result achieved by CryoSpark, but using much fewer like 2D input 2D images. So in the intro time, I will stop here, and I'm happy to take questions.
Questions? Yes.
Yeah. Can I ask a question about the posterior sampling?
Yes.
I guess ideally, like for the accurate accounting for the PY DMXT, you have to integrate of the whole posterior X0 mapping. Just like we have the PX0 DMXT.
Yeah.
But here, like I guess you are doing the posterior sampling by doing the one-step mapping to from XT to X0, like just taking expectation like that.
Oh, okay. Because it's a flow matching.
Yes.
Flow matching is based on like the interpolation between XT and X0. So it's very easy to do this like direct mapping from XT to X0.
Okay.
If you do, for example, like diffusion model, then there's an underlying ODE or XDE, then you need to do integration. So this is due to the advantage of a flow matching.
Any other questions?
Yeah, I guess also a question kind of on the sampling stack to incorporate this likelihood information.
Yeah.
So I know like there are a lot of methods developed specifically on the sampling. Like, how do you maybe like instead of like generate like one trajectory, generate a lot of trajectories, reweight them. There are a lot of methods on that. So what do you see as like the practical difficulty in like actually accurately incorporating likelihood term in the sphere?
Yeah, so in our paper, we actually, for different downstream tasks, we need to use slightly different way to incorporate the likelihood, which demonstrates the trend you mentioned. But on the other hand, I think the fundamental difficulty is because I just illustrated the result of four synthetic downstream tasks. And for all of them, we know the operator A, or we can estimate the operator A from some data set. But in the real world application scenarios, the forward operator or this operation, for example, degrade operation, is not immediately clear. So we have to assume certain physical process that governs this degradation. And this assumption may introduce some additional like modeling error. So no model is correct, every model is wrong, but some model might be reasonable. So we will face the same challenge here because we don't know what's the exact degradation model we should use in different real world scenarios. And this is the part we are trying to tackle in our journal submission, because we started additional experiments on some real scenarios, real downstream tasks rather than the synthetic downstream. And we have tried a bunch of things, and we also made some small progress. So I'm happy to share when the paper is coming out.
That's time for a quick question.
How do you compare like these like voxel-based foundation model versus atomic model-based foundation protein models? Because you can also like find foundation model based on
okay.
atomic models.
Yeah, I think the answer to that is easy because the protein mining model is more similar to GPT. It's more similar to.
I'm not thinking protein. I'm thinking like things like chroma, like diffusion-based. Atomic models.
I see.
Versus like voxel-based models.
Okay. I see. Yeah, like for example, the item like the decoder in AlphaGo 3. Yeah. I think the atomic or all item model is definitely more preferable in many scenarios. But the challenge is if you want to do like all item modeling, then you need to pay more like computation. And that's the reason we go with this voxel or volume-based foundation model. And also the density map itself actually can be naturally like modeled as like a lot of voxels. So probably in this scenario, there's no like strong need to go with the all item model. But for example, convolutional modeling, which also presented in model talk, I think that scenario, the all item modeling might be more preferable because you are essentially estimating the sequence of like dynamic protein structure. And for protein structure, I think the most precise way to model is to estimate the coordinate of each item, and that be the most ideal way to do modeling. Yes.
Thank you, Chen.
Yeah, thank you.